Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Joanna Smart from Business Review, and I will be your host. It's our pleasure to have biopharmaspec with us today, who will be discussing understanding the importance of higher-order protein structure. Today's guest speakers are Dr. Mark Millichip, Technical Director, Physicochemical Analysis, and Christopher Zegenfuss, Laboratory Manager. I'd like to welcome you to our webinar platform on 24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click Submit. We will allocate some time at the end of the session to address any questions or thoughts that you may have. If you click on the survey widget, there are a few questions in there for you to answer in order to provide feedback. You can do this during or immediately after the webinar. If you click on the green resource list widget, you will find some PDFs from our speakers to view or download. Please use the yellow help widget if you require any assistance, and you can move, resize and maximize any of the windows in front of you to get a better view of the slides. But now please allow me to welcome Christopher.
Thank you very much, Joe. It's my pleasure to welcome you all to this webinar on the importance of analyzing higher order structure. My name is Chris Zegenfuss, and I'm the Laboratory Manager of biopharmaceuticals United States Laboratory. Over the course of the next 45 to 60 minutes or so during this webinar, we'll be going over these topics. We'll give you a brief introduction to biopharmaspec if you don't know who we are, and then we'll get into higher order structure analysis, regulation outlines. Then we'll get into the core analytics used by biopharmaspec to assess higher order structure comparability. And then we'll end with a summary and a Q&A session, as mentioned. So for those of you who do not know biopharmaspec, we are an independent, privately owned contract research organization founded in 2014 by Professor Howard Morris and Dr. Andrew Reason. We have two labs, one in the U.S. and one in the UK. Although biopharmaspec is relatively new, many of the senior management and staff have a previous 30-year history working together as part of the MScan group of companies from 1979 through 2010. Our focus as a service provider is the structural and physicochemical characterization of biopharmaceuticals, using state-of-the-art mass spectrometric and chromatographic instruments and methods, along with other supporting technology. Our staff have extensive combined biopharmaceutical experience, much of it based on the application of Howard and Andrew's pioneering academic and industrial research in the field of mass spectrometry. So here we have a brief look at some of the highlights of some of that research over the years. From the late 60s into the early 80s, Howard's academic research included the first mass spectrometric peptide mapping, the first successful protein sequencing by mass spectrometry, and the first MS-based disulfide bridge analysis. Many things which are now commonplace in laboratories around the world were pioneered by biopharmaspec senior staff. Along with those pioneering methods, Howard also introduced high-field magnet mass spectrometry and conceived of the QTOF MS-MS geometry that allows MS analysis of complex biomolecules to the high degree needed for today's applications. Realizing the need for high-end mass spec services in the then emerging biopharma market, Howard established MScan in 1979. Andrew was key in transferring many of Howard's academic research things and applications into the commercial setting in the 90s and early 2000s as a part of the MScan group. Here's a partial list of the molecules that biopharmaspec scientists have characterized over many years of experience. In several instances, our staff worked on these products when the innovators were being developed and now are aiding companies in biosimilar development. And as you'll see, there's a long list of antibodies, EPOs, many of the very popular glycoproteins and such that are being developed for biosimilars these days. And then here we have a little schematic showing the stages of preclinical biosimilar product development. So if we start in the upper left-hand corner, we see defining the primary sequence of the reference medicinal product at the protein level. This is critical to know exactly what it is you're trying to make a biosimilar of. You need to know that sequence very precisely at the amino acid level, and we do this via mass spectrometry along with other techniques such as automated Edman degradation to allow the leucine-isoleucine determination. Then we move on to choosing the best cell clone. Some people in the past had just used bioassays and such to look for a high yield, but we're finding that using high-end testing data to look at similarity of the molecules early on in the process is very beneficial to our clients. During the course of development, it's important to develop and qualify purification methods to remove interfering excipients, such as amino acid stabilizers or sugars or polysorbate. So that some of these things interfere with the characterization process, so they need to be removed, but they need to be done in such a way as to not induce post-translational modifications or other structural changes. So when possible, we like to look at the molecules both before and after purification. Moving along the schematic, we get to determining the primary sequence of the biosimilar and showing that it is comparable to the reference material. And in this case, this is one place where the regulators, we often use the word similar or comparable, but in the case of the primary sequence, it must be identical. That amino acid sequence of the protein of the biosimilar must be identical to the reference product. And then we get into determination of the extinction coefficient of the biosimilar, and this demonstrates that it's comparable to the reference material again, and it helps with quantifying the amount you have in your protein concentration, and this is done using a simple 280 measurement. And then finally, we'll get into comparability assessment using multiple batches of reference material and biosimilar. We do this using multiple tests, examining the primary structure as well as the secondary and tertiary structure. And at this point, I will turn things over to my colleague, Dr. Mark Milichip, Technical Director of Physicochemical Analysis at BioPharmaSpec. So Mark, I'll turn it over to you.
Thanks a lot, Chris. Just like to say welcome to everybody as well from myself. So, as Chris very well and nicely put, this is the basic pathway of a biosimilar product development as we see in BioPharmaSpec. What I'm going to do over the next 40 minutes or so is hopefully take you through the steps that are particularly relevant to higher order structure and try and discuss with you how I see things and where we as BioPharmaSpec see the importance of where you should be doing your higher structure order analysis alongside the primary structure and the physicochemical analysis that Chris has described. So first of all, we're going to start talking about the RMP, which is basically the starting point of your characterization. I think this is important. As Chris said, you have to completely understand the sequence of your primary, your reference medicinal product, but also it's important to realize that characterization actually starts with understanding the RMP. Far too many people consider characterization starting when you first produced your first batch of biosimilar, and then you start to characterize it. Personally, I think that comes in later, but the idea of fully understanding the reference medicinal product, not only for the sequence, but it sets your baseline for the following characterization studies that you will be undertaking with regard to the batches, the different lots, the age, and it starts to allow you to assess what is a critical quality attribute, a CQA, of the RMP that you then have to match and follow for your biosimilar. So it gives you the idea, your playing field of what you're aiming for, and helps you try and understand your target molecule a lot more. And as it says here, the AMA talks about the reference medicinal product and talks about the different ages and different batches and what should be used as your RMP with regards to the specific formulation and lot. So moving on from that, once you start talking about your RMP and trying to match that for your biosimilar to prove comparability, what do you actually have to do? How do you go about characterizing a molecule and making a biosimilar? And the guidelines here, for example, the EMA, talks about it in their specification section. And as it says, for any biotechnology-derived product, the selection of the tests to be included in the specification or the control strategy for both drug substance and drug product, and I think that's quite important, it should be specific to the product and also defined as described in ICH Q6B. And so exactly what is ICH Q6B? Here you can basically split the Q6B guideline or suggestions, advice, down into two sections, into the structural side, as you can see on the left-hand side of the screen, and also the physicochemical on the right-hand side. Now, the structural side is very much aimed at the primary structure, which obviously is very important, but it does also mention some secondary and tertiary analysis, and we'll go into a bit more detail with that as well. And what I think is interesting, what I'd like to point out at this stage, is that these ICH test procedures, for what they recommend, basically very much follows BioPharmaSpec's portfolio of techniques and skills that Chris has already described in the first few slides. And that's not by coincidence, that's actually by design. And we pride ourselves on trying to give you, our clients, what we need. So moving along the biosimilar pathway, we talked about the primary sequence of the RMP, and the next important area is choosing the clone selection. And as you can see, you have a lot of analytical techniques that can be used to obtain the broad structural assessment of the products from a number of different cell clones in a very short period of time. These findings are then used to make the decisions on which cell clone is taken forward, and it's based on product quality supported by the high-end analytical data, again, as Chris mentioned in the past. Now, this can be based along things like glycosylation, glycoproteins, high titer producing, but also, as we're now trying to talk about higher order structure, things like aggregation, or if you have a specific refolding process that needs to be undertaken in either your downstream processing or product specific. So certain secondary and tertiary analytical techniques may also need to be taken into account to try and define and say what is a good quality product or a good quality producer cell clone to take forward. And obviously this very much depends on the CQAs of your target molecule. Just as an aside, to let you know, for BioPharmaSpec, our basic package off the shelf for this would include high-end analytics such as intact mass. We would suggest doing a single peptide map, a single digest peptide map. We would look at oligo profiling, ICIF analysis for charge distributions, and we would recommend doing aggregation studies, particularly for monoclonal antibodies. Moving on to the next step, as Chris also said, is you need to develop and qualify the purification method. This is because buffer excipients can often interfere with the analysis of biopharmaceuticals. As Chris said, there's surfactants such as tween can interfere with the mass spec analysis and also analysis of the monosaccharides. Equally, when you're looking at secondary, tertiary, and higher order structures, chiral compounds can have an effect on the analyses and what you're trying to do with the instrumentation there. Therefore, you might have to purify the active biopharmaceutical ingredient prior to the actual analysis that you're trying to undertake. And the purification method needs to be developed and qualified prior to use. One other interesting fact with this is quite often the formulations of the RMPs are patented, and these patents last longer than the actual patents on the APIs, the RMP themselves. And as a result, you can also have different formulations between the RMP and the biosimilar, and these different formulations can have different effects, certainly on the tertiary structure, with regard to what you're looking at. So one of the advantages of actually trying to develop and qualify the purification method is to actually analyze both the RMP and the biosimilar in the same buffer system, so you are actually looking at a true comparability of those two proteins in the same buffer system. Okay, so the next step doesn't really include higher order structure, but it's here as an aside and interesting because it's the only instance where the regulations actually say the biosimilar and the RMP have to be identical, which is regard to the primary sequence. Everything else you have to prove similarity or comparability, but this is the one section that is very important that needs to be identical. We don't have time to go into this that much today, but back in November, me and my colleagues, Richard Easton and Andrew Reason, gave another webinar on this platform, and please, if you're interested in this, go back and refer to that. And also, I think the link or the webinar is actually on our website as well, so please go back and check those. So moving on again slowly, we bypassed the extinction coefficient because again that doesn't include higher order structure. We're now talking about comparability assessment, and if you like, this is the mainstay of what we're trying to do in a characterization sense of biosimilars. And the whole point is you have to demonstrate that the biosimilar is structurally comparable to the reference material and perform the full structural assessment analysis of the physicochemical properties as outlined in ICH Q6B, but also any relevant product guidelines. The number of batches you need to analyze is product dependent and needs to reflect the variation of the reference material as I showed on the previous slide. This is also where the higher order structure and the comparison comes in. So all this structural, primary and secondary and tertiary data should be used to provide a balanced comparability assessment. You can't analyze just one thing in isolation. You have to try and take everything hand in hand, side by side, and try and make an understood, defined, clear definition of what's going on. And as a result, the assessment needs to be made by a selection of experienced scientists who are relatively experienced in assessing this broad set of data and not just being specific on one or two tests. You also need to understand the experimental variation for each technique that's involved, and therefore you can look for the minor differences and say whether something is significant or not significant and what's inside or outside the expected experimental error. And obviously the structural data does not stand alone. It has to be done hand in hand with the cell-based assay and the functional assays of the product that you're trying to look at as well. And this all very well fits in with the idea of what we call the analytical funnel and the philosophy involved where you're looking at comparability. So at the very top, you have the option of all the possible tests, and then you go slowly down and you whittle that down to, instead of all the tests that you can do, it's all the applicable tests that give good data. And then from there, you can distill out which actual tests give good or characteristic data to help you with your definitions of your critical quality attributes. And gradually, as time goes on, you whittle this down into fewer and fewer techniques which you have qualified and validated until you get down to the product release, which is obviously under GMP regulations, which is separate from the characterization side of things. So as an overview, where we fit in with regard to the biosimilar product development pathway that we've just described, obviously at the top there you have the R&D, which is to do with the RMP characterization, the CQA identification, help evaluate the cell clones, and talk about the biosimilar product development and the various analyses you can do. And also, with regard to preclinical, there are times when you have to do further product characterization of the RMP and the biosimilar, more comparability studies, and early phase force degradation. Now, again, I think it's quite important to realize that characterization doesn't necessarily stop at preclinical. On occasion, you also have to do further work in the clinical phases, which may sound a bit weird, but to be honest with you, there are times when if you just have your 3 GMP batches of your biosimilar that you're trying to match to the various RMPs, that doesn't give you enough data points to actually work in a characterization sense to test whether you are comparable or not. So quite often you will also have to test the batches that you've used for the clinics, phase 1, phase 3 in biosimilar method development, and this will help you give more information and try and give you a greater understanding of what's going on and give you a greater ability to define and decide whether you can call your biosimilar or not. One of the other areas that characterization comes in is with regard to investigations into what happens actually in the clinical trials. Quite often, how you deal with the API or the RMP in the lab when you're just taking it for a test and doing something to it before you inject it onto an HPLC or whatever is quite different from how the clinicians will actually treat the molecule to before they inject it into a person or things like that. And sometimes these differences can have an effect on the structure. And certainly in the past I've done some testing with regard to higher order structure, trying to understand exactly what's been going on in the clinic before they inject it into a person compared to what we see in the lab. So just be aware that characterization is an ongoing process rather than just stopped at preclinical. And I think now is the time for our first poll question. So Jo, over to you.
Thank you very much. That's correct. So, audience members, it's time for our first poll. If you could click the answer that's relevant to you and click submit. The question is, do you think the guidelines are clear and up to date on their requirements for HOS analysis? And the possible answers are yes, I fully understand, no, I think they require an update, or maybe if you know what you're doing. Mark, what sort of results do you think we might get from this?
I'm actually quite interested to hear what the audience say on this. Obviously, I'm biased, and I would personally put myself in Group A. But again, I think you have to understand what you're doing. So for us as a service provider, it would be interesting to try and understand and find out how we can help our clients, whether they need more information from us as when to do these analyses, as well as what the results actually are and how they get them.
Brilliant. Okay, well, we'll have a look at the results now. Okay, so here we can see that 31% fully understand, 38% don't understand and think they require an update, and 31% have said maybe if you know what you are doing. So that's some really good results. Thank you very much there for the audience members for voting. I'll hand back to you now, Mark, for the rest of the presentation.
Okay, thank you very much, Jim. Yeah, that is quite interesting. I think there is definitely a role for us with our consultation side of things as well, if there's a certain amount of people that it's okay if you know what you're doing, which hopefully we're showing to you today. So moving on, and now I want to concentrate a little bit more about the characterization and exactly what the ICH topic Q6B Appendix 6.1 talks about with regard to higher order structure. As we've said more in the past, it's very much focused on the primary structure and the primary sequence with the amino acid sequence, but also the carbohydrate structure and the physicochemical properties. And when you try and look into it in detail, trying to see exactly where higher order structure actually comes in is quite complex. And it isn't mentioned in itself in those terms, and it only comes in when you actually start looking at the spectroscopic profiles, which is the sixth one down in red there under the physicochemical properties. I think part of this is because Q6B came out in 1999, and since then, with the increase in influx in understanding and interest in biologicals, as in new biological entities, but also in biosimilars, there's been a lot more understanding and analysis of what needs to go on when you're trying to look at these biophysical tests. So let's dig a little deeper into exactly what Q6b talks about. And as I said, in the physicochemical properties, under spectroscopic profiles, it says that UV and visible absorbance spectra need to be determined as appropriate, the higher order structure of the product examined in procedures such as CD or NMR, or other suitable techniques as appropriate. So see, you have two specific techniques that are mentioned there. There are other ones available, and we'll go into a little bit more detail about that as the slides continue. But Q6b does, if you like, cover itself, because it does talk about other suitable techniques as appropriate. So let's start off with NMR. As you can see here, this is an image of 3 sets of data of a 1D proton NMR study, which is 1 biosimilar and 2 RMPs. As you can see, the spectra produced are quite complex. However, you have to realize that the midsection from 0.5 ppm to 6 ppm is actually only related to the excipients and buffer related, and is not at all related to the protein structure. The only peaks in these spectra that are related to the protein structure are the very small ones from 0 to 0.5 ppm, and at the peaks greater than 8.5 ppm. The 0.5 to 0.5 ppm belong to the aromatic ring-induced upfield shift methyl-methylene groups of the hydrophobic core, and the peaks greater than 8.5 ppm arise from the backbone amides from the secondary structural elements. And it's only these two real extremes of regions where they represent unique signatures for the protein sequence and secondary and tertiary structures. But taking that into account, you can see that there actually is quite a high degree of spectral similarity for these samples, which suggests good comparability between this biosimilar and its 2 RMPs. Moving on, you can also do 2D NMR, and in this case we generally perform hydrogen-carbon 2D NMR rather than nitrogen-hydrogen 2D, and that is basically because of an increased intensity of the carbon-13 compared to the nitrogen, and as a result there's also more carbon in a protein than there is nitrogen. So again, you're talking about trying to increase your intensities and your signals to try and look at any differences. But what you're seeing here on the screen is basically an overlay of, again, of the same RMP against the same biosimilar against 2 of its RMPs. And again, what you can see is a very good overlay of the spectra, and the only differences that you see here were actually attributed to the differences in the buffer excipients. Now, as I hope you can see, that it's quite a complex set of spectra to analyze and look at, and the main disadvantages with this technique certainly down to cost of the actual instrumentation and maintenance and accessibility to it. To try and get good quality data, it needs to be performed on as powerful an instrument as you can have access to. Certainly over 800 megahertz is what we would recommend, and as a result, the instruments are very hard and very costly to purchase, very expensive to run and maintain, and the data is complex and requires a high level of expertise to actually do the interpretation correctly. Most of these factors put NMR out of the reach of most small and medium biopharmaceutical companies, but certainly NMR is available through some service providers like BioPharmaSpec. Because of the main disadvantages of this, we at BioPharmaSpec would normally suggest that you do and undertake NMR when you first have your first, say, 3x3 study of your 3 biosimilars against your maybe 3 RMPs. But after that, once you prove similarity, you will then go on and use some of the other techniques that I'm going to describe as we go through to continue along with the comparability side of things. And this is, again, fits in with the analytical funnel idea of trying to go from all tests to tests that require to give well-defined analytics and can be used relatively easily and simply. Now, the other method that was mentioned in the spectroscopic profile section of Q6B was CD, and this slide is just to try and make sure we're all on the same wavelength with regard to where CD is. And as you can see, it's down in the ultraviolet region of the light spectrum, and you're looking at peptide bonds and aromatic residue absorbances. So, as I said, CD can be split into two regions of analysis. You have the secondary structure, which is basically looking at from about 170 to 260 nanometers, and that is the far UV analysis. The aromatic region is generally in the absorbance range of 250 to 330 nanometers, and there you're looking at the absorbances of the circularly polarized left and right-handed light of the aromatic residues on the outside of the molecules. And here you can see where basically the absorbances of those amino acids lie. And when you're now looking at the left-hand lower side of the spectrum of the screen, the secondary structure analysis in the far UV, you're looking at the amide backbone absorbances of the proteins, and it was discovered that when looking at this absorbance of the polarized left and right-handed light, certain structures of the proteins gave certain spectral shapes. And here you see the examples of the basic spectral shapes that you see. So you can see a clear difference between an alpha helical protein compared to a beta sheet protein, and also beta turns and polyproline type 2 turns, which are the main areas that you're looking at, or main structures that you can see. So now we're moving on to an example. CD is our primary instrument for secondary and tertiary structural analysis. And what you're seeing here is a far UV analysis of a monoclonal antibody. And here you can see on the left-hand side a very clear high beta sheet content spectra that comes off of the monoclonal antibody, which is as you would expect. Now, one of the advantages, and alongside the visual inspection and interpretation of the data, is you can then input this spectral data into a structural prediction program that is based on a known set of proteins with a known set of structure, and you have an algorithm that searches for similarities between this protein and your base data set, and then comes up with a certain amount of percentages that it thinks is the best fit for the spectra that you've put into the program. And here you can see for this monoclonal antibody, again, you have a high content of beta sheet, which fits and matches with what you expect and what you see on the left-hand side. Now, moving on slightly, now we're talking, these are examples of near UV analysis, and these are two batches of a biosimilar. And as I said, the near UV is more to do with the aromatic absorbance of this circularly polarized light and differences between left and right-handed. And you can't really get so much defined structural analysis on this. You can't say how much is alpha helix or beta sheet, but it gives you a very good, almost like an excellent fingerprint of exactly whether these batches are the same. Because if these overlay in a different way, or you get a different absorbance because the aromatics on the outside of the molecule are in a different orientation or a different environment, then you will get a different spectra produced. And as a result, near UV is excellent for showing similar folding types from different batches of the same product. It also can be very useful when you're doing your early phase force degradation, because quite often you actually lose your tertiary structure before you lose your secondary structure. And as I said, as a result, it's useful in force degradation studies because what you're trying to do there is prove that not only are your molecules similar in the shape and function that they have, but also in their degradation profiles of how they fall apart. And again, that gives you more confidence in the fact that your molecules are actually the same or similar. So, in summary, CD is an excellent informative tool. It's actually named in the ICH guidelines. So, again, it's almost like a must-do to do. It's relatively readily available technique for both secondary and tertiary analysis. It's a non-destructive method, so you can take it out of the cuvette and use it or use that sample for something else. So again, that's very useful to try and do. Some sample losses are observed when you're doing the far UV analysis because you're using such relatively small amounts and in indemountable cuvettes, but that's a low amount of sample and can be used for batch-to-batch reproducibility, stability studies, and force degradation. And I have to say it is a relatively easy instrument to use as well. Now, moving on, what we were talking about with regard to other techniques as appropriate, and FTIR, Fourier transform infrared, is we're now moving to the other end of the light spectrum, if you remember the slide from CD, and infrared is, as I said, at the other end of the spectrum. And basically, you're looking at the amide 1 and amide 2 are the most informative bands, and these are based around the stretching vibrations of the carbon and nitrogen-based bonds. These can help you provide the qualitative and quantitative information of the secondary structure of proteins like alpha helix, beta sheet, beta turn, and disordered structure or random structure. Very similar to CD, but as I said, it's orthogonal, so it's not exactly the same, and it uses a different set of analytics to try and understand and look at your method. So in a scientific viewpoint, using orthogonal methods is very good because you're using different aspects to try and probe the same sequence structure to see whether you're getting similarity or not. Now, here we have an example, and this again is a monoclonal antibody, looking at it with ATR, FTIR, and this is 3 examples of the same monoclonal antibody analyzed in the same session. And as you can see, you have the main highest peak on towards the left there is the amide 1, and then the one next to it at about 1550 per centimeter is the amide 2. And those are the main bands that you're looking at when you try and assess your similarity for secondary structure. Now, very similar to CD, you can also then take these spectra and put them into a secondary structure prediction program that also uses an algorithm and also comes up with structural predictions of the best fit that it sees for the data. And as you can see here, this monoclonal antibody also comes up with a high content of beta sheet, which is, again, what you would expect and what you can see. So, in summary, FTIR has quite a lot of advantages. It can be used for a variety of physical states, not necessarily all of them related to biologics, but you can use them in solid, vapor, liquid, or semi-solid states. It's easy to operate. There are no background fluorescence problems or light scattering or protein size issues, and can be used over a broad range of concentrations. And is, on the whole, mostly buffer-friendly. You have to do buffer subtractions, but on the whole, most buffers are okay in it. As with all things, there are certain disadvantages as well. It can have water in solution has a strong absorption in the amide 1 region, which can compromise some of the techniques and the subtractions you're trying to do, and also low protein concentrations, less than 5 mg per ml, can be quite quite difficult because of in transmission because of the technique of what you're trying to do. However, there are various advances in attenuated total reflection, FTIR, which is ATR analysis, and some of the data assessment programs are getting better and better with their predictions. So the minimum concentration that you need is being lowered all the time. One of the other advantages that FTIR has over CD is that it's not as affected by the exaggeration of the alpha helix absorbance of a molecule. So they are FTIR is quite good for molecules that either have a mixture of alpha helix and beta sheet or have a very low amount of alpha helix in them in the first place, which is such as monoclonal antibodies. And again, I think this is a nice example of how things have progressed from the recommendations in the Q6B with regard to what is the best technique to try and undertake for certain analyses. And we recommend trying to do both FTIR and CD side by side. Now, the other technique that is actually mentioned in Q6B but not under the spectroscopic profiles section is fluorescence. And these are examples of intrinsic fluorescence where you're looking at the fluorescence of your aromatic residues once you excite them, such as tyrosine and tryptophan, and the emission profiles that you see along the bottom of the screen there at these different excitation wavelengths give you an idea of the local environments that these tryptophans and tyrosines are in, and if they are in the same local environments, then you expect to have the same emission spectra, and therefore you are inferring similarity. As I said, it's been quite superseded more these days with things like techniques like CD and FTIR, but due to the sensitivity of the method, I think it's something that people still do and expect. And again, I would suggest that it's something that we do at the start of the analysis, and it's then to be considered if it can be dropped at a later date. You can also perform extrinsic fluorescent spectroscopy, which is related to using fluorescent dyes and binding to hydrophobic groups of the protein, and you do, if you like, a titration of the amount of protein to the fluorescence dye, or the titration of the fluorescence dye to the amount of protein. We don't really have too much time to go into much detail about that today, so if there are any questions with regard to that, we can certainly help you and follow up after this webinar. And Joe, I think we're now time for another poll question.
Yep, that's right. Thank you very much. So audience members, it's time for our second poll. The question is, which methods or instrumentation do you currently use to assess HOS for secondary and tertiary structure? If you could click the answer that's most relevant to you and click submit. The answers are CD only, CD and FTIR, CD and NMR, CD, FTIR and fluorescence, or all of the above. What sort of results are you expecting to see from this, Mark?
Thanks, Joe. Again, I'm quite interested to try and see what the audience feels it does at the moment, and whether it sticks to, if you like, what the Q6 regulations say with regard to just using CD and FTIR, or whether they are, CD and NMR, or whether they're actually looking at trying to use whatever techniques are available or as appropriate to try and understand which is the best. So it's trying to gauge and understand what the audience currently see as their mainstays.
Okay, brilliant. Well, let's have a look at the results. So here we've got CD only with 12%, CD and FTIR with 16%, CD and NMR with 8%, Majority here, CD, FTIR and fluorescence with 36%, and then 28% with all of the above. So thank you very much to the audience for participating in that poll. I'll hand back to you now, Mark, to continue the presentation.
Thanks, Jo. Yeah, that's quite a mixture. I'm quite intrigued by that, but it's good to know that CD, FTIR and fluorescence are all being used, and also then in second place, all of the above, so the NMR as well. So now I'm going to move on. I've talked a lot about secondary and tertiary structure. What I want to talk about now is aggregation. And maybe some people are sitting there thinking, why is he now talking about aggregation? How does that fit in with higher order structure? Now, basically, aggregates can be described as quaternary structure. So it's molecule to molecule interaction. And this is where higher order structure and aggregation comes in, and you need to analyze both of these side by side because they are basically the same thing. So this is why we are now starting to talk about quaternary structure or aggregation as we're talking. So again, in the initial part of Q6B, it didn't talk about quaternary structure at all. And the only place aggregation comes in the Q6B is in Appendix 6.22, where it talks about the product-related impurities, and there it talks about including degradation products, and this is where, in Part C of that, the category of aggregates is included in a potential degradation product of your molecule. And as it says here, you need to use certain techniques, but also it then says as appropriate analytical procedures. And here you see things like site exclusion chromatography and capillary electrophoresis are mentioned by name. Now, on the image and on the right-hand side, you can see two of our scientists with our SEC-MOLS system that we use routinely in biopharma spec. Now, moving on, so we recommend using SEC-MOLS as our mainstay of our biopharma spec-based aggregation analysis, and this involves looking at a 3 detector system coming out of an SEC column, and these are UV refractive index and multi-angled light scattering detectors. We recommend this, and so do the regulators, because they become aware of this, because as it's become more and more aware that if you have larger aggregates, instead of just absorbing UV light, they tend to scatter the light rather than only absorb it. So SEC-UV on its own potentially can underestimate the amount of large aggregates that you have in your system. So this is why it is beneficial to, if you like, back up or qualify your SEC-UV data with SEC-MOLS to try and prove that your SEC-UV data is true and real rather than masking or hiding any further analysis. In the image, you can see here along the bottom is the time that the peaks eluted. The blue line is the UV trace, where you see the main monomer peak and then two earlier eluting peaks that come out as shoulders that aren't clearly resolved. And in this case, when you look at the red dotted lines, what you're seeing is the output from the MOLS detector, and here you have clearly the monomer, the dimer, and for the first peak, as you come off the most smallest one, we're not actually getting a MOLS reading, and this is because we've actually reached the limit of separation of the SEC column that was used. So this isn't just a trimer or a tetramer, this is a mixture of multimeric compounds, which is why you're getting quite the dotted line that disappears up into the sky and off the scale. So SEC-MOLS is something we use and we recommend to our clients that you have to do and qualify to make sure your SEC-UV is working correctly as you do it. Now, There is also an orthogonal method to SEC-moles, and this is SV-AUC, which is Sedimentation, Velocity, Analytical, UltraCentrifugation. It has become aware and known that due to certain properties of the SEC method, where you inject quite a small concentrated sample at the top of the column and inject that onto the column, that sample then goes through quite a large dilution effect and undergoes lots of shearing and turbidity forces before it reaches the detectors. And it has been seen that if you have non-covalent interactions and attractions that can still form aggregates, SEC-moles and SEC can disrupt these and take them and pull them apart. So again, you don't actually see the non-covalent interactions and aggregates that you're seeing. And certainly the FDA has recommended, therefore, because you need to do not only a column-based aggregation analysis, but a column-free aggregation analysis. And SV-AUC really comes into its own with regard to doing that. And this is something I'm going to discuss and give you a little bit more detail with regard to what is actually going on in SV-AUC. So here, it looks quite complex, but it is relatively simple apart from when you get to the equation that you're trying to define what's going on. So at the top, you have your homogeneous mixture at T0, and you apply your centrifugal force, as the arrow suggests, out towards the right-hand side of the screen, with your radial distance of the centrifuge increasing from the left to the right. And say after, where it's T90, you actually have a higher concentration or proportion of material at the bottom of the cell compared to the top. And the rate and how much pellet is based on certain known factors, such as the density of the buffer that you're pushing the sample against, also the space filling, the partial specific volume of the molecule of interest that you're trying to separate, also the number of species that are present, like the number of aggregates. If you have a higher number of aggregates, they will pellet faster because they have a higher mass, a higher molecular weight, so they will go down quicker. And as a result, as I said, it's quite a complex equation with regard to describing this process, and this equation is the Lamb equation. So basically, you can rewrite this and reorder this, and you're looking at the difference between the chemical potential, which is, if you like, what keeps samples in solution, repels them, keeps them from each other, and the sedimentation potential, which is, if you like, what attracts molecules to each other, and then they can degrade that way. Obviously, the sedimentation potential is also affected by the centrifugal force that you're put on it. And in this case, when we're talking about sedimentation velocity, we have a higher sedimentation potential than we do a chemical potential, which is why the molecules pellet at the bottom of the cell. Now, moving on, this gives you an idea of the data output that you get from SV-AUC. And this example is BSA, and we've chosen that because I'll explain a little bit later. But it's to do with, clearly you can see a nice amount a high amount of dimer and trimer, as BSA is known to have in solution. But basically, the top graph there can be split into three, and the very top is actually looking at the absorbance profiles, taking at different times as you go down through the SV-AUC run. So these ones at the left-hand side are the early absorbance traces, and the ones on the right-hand side are the later absorbance traces. The middle section is more to do with the residuals and, if you like, the quality of fitting of the SEDFIT program that you've got. And then you can convert this absorption set of profiles data with time against the known factors and the parameters that you've put in with regard to buffer density and this kind of thing. And you eventually come up with a sedimentation coefficient value. These can then be converted to masses, which again can give you an indication of how good a fit you've got. But equally, it shows you really quite nicely the amounts of monomer, dimer, and trimer, as I said, in this case in BSA. And the red and the green are, the red scale is the expanded scale to increase and show you the dimer and trimer content higher. So now trying to put that into context of biosimilar development. Here we're showing an example which is 2 RMPs against 10 different batches of biosimilar. And here you can see that the monomer peak is just around about 6 to 6.5 S, which is what you would probably expect for a monoclonal antibody. But also they overlay really, really nicely. And then if you look at it on the expanded scale, here you're seeing also quite a lot of similarity in the amount of dimer and trimer there. So not only does it work on BSA, but it works in real samples as well. So again, Joe, I think it's now time for our final poll question.
Brilliant, thank you very much. So this is, as Mark said, the final poll question. Audience members, if you could choose the answer that's relevant to you and click submit. The question is, which of the below techniques have you used for aggregation assessment in the past? SEC-MALS, SV-AUC, FFF-AF4, SEC-MALS and SV-AUC, SEC-MALS and FFF-AF4, or all of the above? Mark, what do you think the results might show?
Thanks, Joe. Again, this is a bit of a fact-finding mission on my side of things. As Chris and myself have said all the way along, we do try and keep ourselves at the forefront of instrumentation, and we know that field flow fractionation is, if you like, an up-and-coming technique to try and understand and define aggregation profiles, and we are just trying to gauge how much interest or need our clients and potential clients have with regard to this new technique.
Okay, great. Well, I'll just have a look at the results then. Okay, so here we've got 48% using SEC-MALS and 32% using SEC-MALS and SV-AUC and 20% using all of the above. So thank you very much to audience members for participating in that poll. Mark, I will hand back over to you to wrap up the webinar.
Thanks, Jim. So yes, I just have 2 last slides now. So in summary, we at BioPharmaSpec consider ourselves highly technical and collaborative partners for your outsourced biophysical structure and physicochemical requirements. We use state-of-the-art instrumentation and have a pioneering approach to methodology. We have a thorough knowledge of the ICH Q6B and its product-specific applications, and we have a comprehensive experience of a wide variety of products, over 150 biopharma products, including more than 50 biosimilar development comparability programs. And as Chris said, we've been around long enough that we've seen a lot of these biosimilars that are coming through now, and we certainly helped define and understand them when they were innovators in the day. And lastly, I would like to say that we consider our consultancy services as a standard and try and help put our data into context within the rest of your development program. And my last slide is just our contact details with regard to myself and Chris, and also our salespeople of each region in the world.
Brilliant. Thank you very much, Mark, and thank you to Chris as well. Unfortunately, we've not got time for any questions, but we have had a lot of interesting questions on the slides and techniques such as HDX, MS, DLS, and primary structure methods like Edman chemistry. And we just want to let you know that although we don't have time to answer those questions on the webinar, a BioPharmaSpec scientist will get back to you as soon as possible. So just keep an eye out for an email from them. So that just leaves me to thank Mark and Christopher for what was a great presentation, and to thank BioPharmaSpec for sponsoring this session. To the attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter @BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you once again, and I hope you all have a lovely day.